Okay, so what is being carried out in DNA amplification? Mini chromosome carrying this gene of interest is taken out. It is identified, it's taken out, it is isolated, and is introduced into a self-replicating chromosome, which can be of smaller organisms, unicellular organisms, or prokaryotes. Usually we take that is yeast artificial chromosome, bacterial artificial chromosome, phage 1 artificial chromosome, etc. That is, we call it as yak, backpack, etc. So in the or phage vectors, viral vectors, etc. Right? So the circular DNAs are always preferred, and to the circular DNA we insert into it our gene of interest. So it is having two different genes coming from two different organisms. So we call it as a chimeric gene. It's known as a chimera. So chimera means a typical example for chimera or mythology like like Narasimham. Right? Narasimham is having the head of a simham and a lion and a body of a man. The same. So it is belong to two different species, right? So the same thing is we are implementing in our molecular technology also, and we call it as chimera. Chimeric gene. So such a chimeric gene is produced, and this chimeric gene is now introduced into a vector. Vector means vehicle. So usually we uses viruses, right? So this viruses then is introduced into a our animal, we usually use guinea pigs or rats, right, or monkeys or rabbits, etc. So we introduce it to it, and what does the virus do? Virus incorporates. Now the virus is having this chimeric gene. So this chimeric gene now gets along with the host cell, right? So short strand of DNA that are complementary to DNA sequences on either. It is ready for amplification. This is how we are doing in the in vitro process. So the main enzyme responsible for carrying out this amplification is known as DNA polymerase. Polymerase means, what do you mean by polymerization? It's multiplication. So the multiplication is enhanced by an enzyme which is known as DNA polymerase, and one of the most important tool we use in molecular biology, DNA polymerase, right? And this the very same enzyme we are making use of in PCR also. PCR, that is polymerase chain reaction, that's the expansion of PCR. So in PCR also, one of the most important tool we make use of is a DNA polymerase, which is responsible for amplification. So how are you carrying out the amplification? We have got only a single gene, and from the single gene we have to study, and you have to correct it. You have to make corrections also, right? So we have to amplify your single gene. So that is being carried out by a technique we call it as a PCR. So PCR is one of the most advanced technique. Before PCR we were using of RFLP techniques, that is restriction, fragment, length, polymorphism, etc. It was a technique which was earlier used before the invent of PCR. So PCR, this technique was developed by a scientist named Kary Mullis, and he developed in the year around 1993. He was a chemist, in fact, and he was a person to develop this technology known as PCR, and he was awarded a Nobel Prize in the year 1993. Of course, he received the Nobel Prize in 1993. He has developed PCR earlier, right? And he was for this work he was awarded with. He was given Nobel Prize in the year 1993. So what is being done in the PCR? PCR is a very useful tool which has deleted all the drawbacks of all the earlier techniques. So PCR, again, one of the most important advantages is that it requires only a small time, and within a small span of time you can have numerous number of the gene of interest. You are creating numerous number of genes from a single gene, right? That's the advantage of PCR. And you need only a single gene, and very single gene will be amplified to around 10,000, 25,000, etc. within a short span of time. That's the main advantage of PCR, and again it is not a time consuming process. It requires only very short time, right? Again, PCR are of different types, and in the case of recently we are using for the COVID-19 etc., we are using the RT-PCR, right? So there are different types of PCRs. What is being the basic technology is that the basic principle behind is that we are having our gene of interest, right? This single gene, this is our gene of interest. See, so it's a short strand of DNA. It is allowed to first split into two different strands, right? The coding strand and we call it as a, this is known as a coding strand, this is not a non-coding strand. അങ്ങനെയൊക്കെ template strands ഉണ്ട്, right? So this is a DNA template strand. So this double-stranded DNA is first cut into two single-stranded DNAs by we are just heating it for up to a temperature of 95 degrees Celsius for 15 seconds. The too short time. Within 15 seconds, if you are heating the DNA for about to a temperature of 95 degrees Celsius, it will be getting split up into two strands, two individual strands, and this is the template strand. That is, this is the direction in which it undergoes doubling. DNA doubling happens only in the 5' to 3' direction, right? So that is why we call it as a DNA template strand. This is the non-template strand, right? So these are the two strands. Now these two strands, this strand is subjected for annealing them. Annealing means this is a single strand. No, this single strand is again undergoing, it is making its complementary strand. It has to prepare its complementary strand. So the complementary strand is prepared by adding DNA primers. We add short sequences of DNA to it, so that only those sequences which are having a complementary nucleotide pairs will be getting pairing with this strand, right? So the pairing starts from this direction, right? So these are the primers, DTTP, DCTP, primers. We are putting some primers into it, and those primers which are complementary to this template strand will be going and getting annealed to it. And this annealing require again only around 35 seconds, a small span of time again. And then what happens? This is a the annealing requires a temperature of around 55. So from 95 degrees Celsius, you have to reduce the temperature to around 55 degrees Celsius, right? So the temperature is reduced to 55 degrees Celsius, and at this temperature, what happens? It starts pairing. It makes a double-stranded DNA, right? So both the strands undergo what? Pairing. So according to base pairing rule, both the strands undergoes pairing, and they give rise to two individual DNA strands, right? So these are the two strands. Now we are having two different strands. So this is how the polymerization happens. This happens in this direction, and this it requires some time because it's in the opposite direction, right? So the two, we were having only a single strand. Now it is after a short interval of time, what happens? It becomes twice. Now these two strands are again subjected for the next round, next cycle, right? So next time we'll be having what? How many number? This strand produces 2. This strand produces 2. So 1, 2, 4, then 8. So it is progressing in the form of geometric mean. So you will be having numerous number of, within 24 hours you can have 25,000 to 35,000 of this gene of interest. So that's the advantage of PCR. You can amplify it to a number of times within a short span of time, right? And the enzyme we are using is a DNA polymerase enzyme. That is, it is the DNA polymerase enzyme is primer, but it's not necessary. It is not sufficient for the DNA strand to be supplied with the primers. The primers we are putting, but the primer in order to undergo polymerization, what should be done? You have to add a tool which is known as DNA polymerase. So that is why the PCR is highly dependent on an enzyme, or the most important tool which is making use of in PCR is a DNA polymerase. right? So this DNA polymerase, in the presence of only the DNA polymerase, it can carry out the amplification. It can form double-stranded DNAs. These double-stranded DNAs, again, it is subject for the next round of amplification, so that it will be producing the products in a geometric fashion, not in an arithmetic fashion. Okay, so there are different, I've told you that there are different types of PCR. The conventional PCR, it requires a much more time when compared to that of the recent advances. The most important types of PCR, of course, one is a RT-PCR that we are making use of now, right? For the viral infection diagnosis of the infectious diseases are diagnosed by means of RT-PCR, that is reverse transcriptase PCR or reverse transcription PCR, RT-PCR, that is we are using our mRNA. That is a material used. The raw material used for the amplification is the mRNA, not the DNA, and it is a widely used PCR, the widely used type of PCR, again for the diagnostic purposes, right? Then there is a asymmetric PCR, inverse PCR, then nested PCR. It is having much more sensitivity and specificity when compared to that of the others, because they are using a more specific set of primers, right? Then there is in situ PCR amplification, etc. So these are the different types. The RT-PCR, nested PCR, inverse PCR, asymmetric PCR. Then another one is a real-time PCR. It can get confused with RT-PCR. RT-PCR is reverse transcriptase PCR or reverse transcription PCR, which makes use of reverse transcriptase enzyme with the help of, that is mRNA to be converted into DNA. So the conversion of RNA to DNA is made possible with the enzyme known as reverse transcriptase. So that is why that PCR is known as RT-PCR, reverse transcriptase PCR. There we are using mRNA as the genetic material or the raw material we are using. So another type of PCR is a real-time PCR, or it is also known as quantitative PCR, or it is also known as qPCR. So it should never be confused with the RT-PCR. RT-PCR is not real-time PCR. It is a reverse transcriptase PCR. Right, so RT-PCR is considered as a gold standard for the rapid diagnosis of viral infections, and that we are doing now. Right, and then the qPCR is the best out of it is more precise. The real-time PCR, it's much more specific and it's much more accurate. It could, again, more reproducible than that of RT-PCR, but the most widely accepted or the most common one is the RT-PCR when compared to that of the qPCR. Now, what are the advantages? It eliminates the need to clone the sequences by in vivo replication. Again, it is happening by means of geometric fashion. Right, again, thermal cyclers are there. That is, we know that enzymes, most of the enzymes are proteins. So it is natural that when the temperature is elevated, these enzymes can get denatured. So denaturation of enzyme can happen. But the PCR, RT-PCR is employed with certain instruments like thermal cyclers, in which the temperature is adjusted in such a way that the enzymes won't get denatured. Right, again, it undergoes automatically the temperature conversions will be done. You don't have to manually regulate the temperatures. It will be according to the steps it is undergoing, the material is undergoing, it automatically shifts to the temperatures. So they are having thermal cyclers. But the main disadvantage is that the nucleotide sequences flanking the gene of interest is no. Only if you know that is what you mean by flanking sequences, is that if you're having, suppose the human insulin hormone, the gene coding for human insulin, it's having 50 base pairs. Suppose you cannot take out the 50 base pairs as such. It should be provided the insulin gene, it is to be introduced or inserted, should be flanked with a few set of nucleotides on either side, so that the gene as such will be confirmed. That 50 base pairs will not be affected, right, due to some enzyme action. So always these nucleotide sequences, this gene of interest should be flanked with, it should be having a it should having a shield, and this shield should be a set of nucleotide base pairs which will be flanking the gene of interest. So only if you know the flanking sequences, nucleotide sequences of the gene of interest is known, then only you can carry out the PCR. That's only disadvantage